Hi, everyone. Thanks for joining the webinar today. This is Tianjun. It's the Cytobank Bootcamp Session 3, and we'll be focusing on the dimensionality reduction algorithm viSNE in this session. You have seen this before. Cytobank, as a cloud-based platform, is specialized on single-cell analysis solution for high-dimensional data, leveraging the power of cloud compute for machine learning algorithms. We offer structured content management where you can securely save and archive your analysis. You can collaborate and share easily with your colleague via a share button. So the platform is designed for biologists with friendly user interface. We also offer professional bioinformatic consulting service for customized work. In our last session, we talked about the importance of data QC. The algorithm is as good as the data quality you put in. So we want to have a high quality data and perform the necessary data cleanup process before the algorithm runs. Once you get a clean quality data, you can try those algorithms to explore your data and get quantitative results. Here's a typical workflow where you can use viSNE as the exploratory data analysis tool and then run clustering algorithm to get more quantitative results, or run Citrus for group comparison. And best of all, the workflow of run clustering algorithm or Citrus on viSNE can enable you to visualize the clusters on the viSNE map and take advantage of both clustering and dimensionality reduction tools. And today we will be focusing on the viSNE part. We will introduce the basic series showing you how to start a viSNE run and how to interpret the results in Cytobank. So tSNE stands for T-distributed stochastic neighbor embedding, and viSNE is the same algorithm as tSNE, but with improved visualization for cytometry data, and it can run on more events with a downsample step. It is a dimensionality reduction tool that creates a two-dimensional view of high-dimensional data. It plots the data on this two-dimension plots, tSNE1 and tSNE2. Those two channels are arbitrary, so they serve as a purpose of dimensionality and has no biological meaning. viSNE groups the events based on the phenotypic similarity of all the markers via a process called iteration. So each dot here is a cell or an event. The cells close to each other have similar biological expression, then the events far apart. You can also color the dots by a third channel. In this case, the CD8 expression is observed within the group of cells. The expression level is color-coded, making it easier for scientists to not only visualize, identify interesting and rare biological subsets, but also to gauge on those single-cell events across different samples. So here is an animation showing how viSNE works. Basically, it calculates the pairwise similarity matrices for all data points on high-dimensional space, then calculate the matrices in the low dimension. The algorithm then tries to minimize the differences between those two by adjusting the cell point in the 2D space via this iteration process. So as we saw that the cells start out as one group and then scatter out to find the best position on the map. So with each iteration, it minimizes the differences between the matrices until the best 2D representation is achieved. And viSNE also tries to preserve both local and global structure. And the local approaches seek to map nearby points on the high dimensional to nearby points on the low dimensional. And the global approach mapping nearby to nearby points and far away points to far away. So for viSNE, the individual cells can be identified and it can be used to identify small subsets. So for viSNE, in order to identify the phenotype of each island, it can be manually gated by using color by channel. Here we gated on the major subsets using the canonical markers. The marker association can be visualized on the same map. Or you can use clustering algorithm like FlowSum and SPADE on viSNE or viSNE identified subsets. However, viSNE is stochastic. So that means different runs of the same data set can produce different viSNE map. But don't be panicked. That doesn't mean the viSNE is not reliable, but rather the data structure will be very similar with each run. Just the island position on the map can be shifted. And this can be controlled within Cytobank by setting the same seed with the same data set. And we will get to that in much more detail later. So the goal of viSNE is to find a low dimension visualization that best reflects the data structure in the high dimensional space. Like here, both are the 2D representation of your hand, and one is a better representation that reveals all the details of your hands, and the other one is a lot harder to interpret. The same applies to viSNE map. So what is a good viSNE map? As the dimensionality is reduced from high dimensional to 2D space, potentially many maps can be generated. The question comes to what is a good representation? And here on the left is a well-defined viSNE map with each island well separated. And on the right hand here, everything's smeared together, which could be an indication of not enough iteration. If we stop the iteration in the middle, you're not generating a good map. So we have a way to find out if you have enough iteration for your run by looking at a value called KL divergence. And we will touch upon that in the app demo later with the viSNE run. Here is an example showing what the viSNE map looks like for a 20-color Cytoflex LX data. So the viSNE is run on gated live cells, and then 17 markers was selected as the viSNE channels. You can see here this is one file, and each map represents a channel expression on that particular map. So on the first map here, we are viewing CD3 channel on the map. The channel expression is color-coded. So the red islands here showing the CD3 positive events. And the same here, this region is CD8, and this region is CD4, and CD19 positive islands here, then CD33 positive islands down here. We can also gate on the viSNE map based on the channel expression, so we can find viSNE identified CD8 positive cells, viSNE identified B cells, and we can also to compare the viSNE identified island to your manual gating. So this is a comparison between my manual gates versus viSNE identified populations. So this is a map we saw earlier. You will see where is the CD4s, CD8s, and the B cells and the NKT cells. And this is my manual gates in those color blocks. If I overlay my manual gates on the viSNE map, you can see it matched pretty well, with each of my manual gates laid at the same place where viSNE identified population are, like the pink CD4 and the brown CD8. However, if I separate each manual gate and check on the map position, you will see some interesting stuff. So let's focus on the monocytes here. So this monocyte is my manual gate, and I want to see where my monocytes is on the viSNE map. If you remember, here's a viSNE identified monocytes. So you will see this region is a viSNE identified monocytes, and the majority of the manual gated monocyte in this region. However, I still have a lot of other events scattered around on the other place of the map. And why is that? So those locations are where the lymphocytes are on the map, and most likely that when I gate on my monocytes, the gate was a little bit too generous and included quite some lymphocytes, and that's where they come from. So let's look at another case here, NKT cells. If we remember, this is the viSNE identified NKT cells, and if I overlay my, visualize my manually gated NKT cells on the map, you'll see majority of them overlay nicely with viSNE identified NKT. However, I still have some of the events scattered around in the lymphocytes region. It's an indication that it's not, my manual gate NKT is not very clean gates. I should have done CD4, CD8 negative gates before I approach my CD56 positive and CD3 positive NKT gates. This is an indication of how inaccurate and subjective of the manual gates can be. I believe many of the people here might have done the same thing as what I have. Here's another example of how viSNE can be used to answer your scientific questions. This is a paper published by Shram Kudasky on the Blood Journal. So the viSNE map was used to identify Treg subsets between patients with different responses to treatment. Some are responders and some are non-responders. So the viSNE was performed on the CD4 T cells. The highlighted region here is the Treg subsets, indication of CD25 positive, FOXP3 positive, and CD127 negative. There are two subsets of Tregs in this region. One is A and B. There are two subsets of Treg within this region, A and B. When you look at the map of patients who did not respond to the drug treatment, they are high on A region, while in the responder patient, the B subset is more elevated. And when we check on the B subset, it shows more memory and activation phenotype, which is an indication of why it's responding to the treatment. So with those information, we can identify the differences between responders and non-responders and providing valuable information to the research. Here I'm showing you another visualization tool we can use is to run flow sum on viSNE and then visualize the flow sum metacluster on the viSNE map. So on the left here, the flow sum identified metaclusters. They overlaid on the viSNE map with the tSNE1 and tSNE2. You can identify each region and the phenotypic of those particular clusters. Another way to visualize the same data is to use a heat map. You can view the metaclusters of each channel on this heat map. So for our next session, Stephanie will show you the flow sum clustering algorithm next. So now I would like to move on to the live demo in Cytobank. I will show how to set up the viSNE analysis and how to interpret the results using a demo dataset. It's a demo dataset we are going to use today. It's a 10 patient with 2 time points, 1 acute infection and 1 during the recovery phase, with a typical immunophenotyping panel with some function marker and activation markers within the panel design. So we already talked about compensation, scale transformation, and gating tools last time. So today we will go straight to the advanced analysis and start the viSNE run from here. So in order to start a viSNE run, we click on the advanced analysis and start a new analysis. I often give the new analysis a name. I often give by date or some other informative information here. You can select OK. This will bring you to the viSNE setup page. So the first thing is to select a population. The population showing up here are the population you manually gated in the gating window. So in this case, I'd like to select the population as my CD45 positive. So I run the viSNE on the CD45 positive, and I want to run the viSNE on all the FCS files. So it will be 10 patient, 2 data points, total 20 FCS files. Then start to fetching the events. The next step will be select the event sampling. We have 2 options to select the event sampling, either by proportional or by equal sampling. Some of the mistake people often run here is that they run the viSNE on too granular. So in general, we want to start with the higher hierarchy population first, and we can narrow down to a specific subset as needed, but we want to do an exploratory on all the data set before we narrow it down. So the proportional sampling will take the percentage of cells that equals the percentage of the population of the total events. And the equal sampling takes the same number of events from each population or each sample. So whether to use proportional or equal sampling will depend on a few factors. Equal sampling is a good choice for most applications if you're unsure. So using equal sampling makes a visualization of differences in the structure and relative abundance of population across files easily. However, when 1 or more files in your experiment has low event counts compared to most of the other files, that will limit the max number to be included in the run. So in this case, you may use proportional or just exclude the samples with lower events from the run. So the next step here is to choose viSNE channels. Those are the channels that will be used to form the viSNE map based on the expression similarity. Normally, we choose surface markers that will contribute to separate the cell population. So I'm going to search for the CD markers. That will be helpful in this case. Since we already use the CD45 as our gating marker, we would like to exclude that from our channel run. Other markers like scatter channel variability marker, so shouldn't be included. And if you run viSNE on the pre-gated population, like here, CD45 positive, then the CD45 marker should not be included as well. Also, another important note is that you should not mix linear and arcsign in the viSNE run. And we talked about the compensation and the plot scaling last time. So make sure the data is well compensated before we go to the viSNE setup, and scale transformation is also very important for viSNE. Then down here is the advanced setting where we can set the seed, iteration, perplexity, and theta. We talked about we can optimize the iteration to get the best scale divergence value last time. So we can, 1,000 iteration is a very good starting point to start with. And then the perplexity is the rough estimate of how many close neighbors each event have. 30 is also a very good default starting point. If your data events increase or the data panel increase, then we should increase those values properly. And then for the seed, it's random. Once we run the viSNE from here, and the seed, as the viSNE algorithm is stochastic, meaning that it uses random steps to help it to do the initial seeding. So we can set the seed to be the same for different runs to allow us to repeat the same viSNE run on the same data and get similar viSNE island. So setting the seed helps to keep the viSNE map similar between different runs with the same samples. And now we can select run. Once you click on run the viSNE, it will send a request to the virtual machine on the cloud. They will pick up the request and start the analysis from there. So once we send the request, we can close out this browser, or just close the computer. Even we can close your computer and take a break here. You will get an email notification when the analysis is done. Since here we are live, we want to, I will use a already finished run to illustrate the result setup. I know you must have a lot of questions by far. Make sure you send your Q&A question via the Q&A window, and we will get to that by the end. So now I will go to my already finished viSNE analysis with a similar setup. You can get to your viSNE results by either go to the advanced analysis here, or you can go to the action and view summary from the summer linked experiment session in the summary page, or you can get to that from the email notification you got. So I will go to advanced analysis and select on a previous run viSNE. To show you here, you can access your viSNE settings. So if we click on this page and bring up the viSNE setting, it will show you the settings we set up earlier, the population you run the viSNE on, the event number you have choosing for your samples, and also the channels you have selected. And also the advanced settings, you can see those settings in the advanced run. So down here are the analysis name. As we said earlier, you can give some more detailed information of what the population was selected, what was the iteration you set for the selected markers. And then down here at the results page, it will show you the viSNE run in how many hours, with a KL divergence value of 5. So we can go to the generated results by click on this icon here. So that will bring you to the experiment summary page of your viSNE results. And down here in the notes section, it shows you the, in the comments field, which shows you the runtime and the KL divergence value at each iteration. So as we saw in the slides earlier, the iteration 1, you have a really, really high KL divergence. I mean, the differences between the high dimensional and low dimensional space are dramatically different. That's the initial CD. With every 50 iteration, the value tend to drop down, and you can see by the iteration of from 700 to 750 iteration, the value drops dramatically to a single digit. And then as you increase the iteration, you get a little bit better, but not a lot more. So for this example, I used a 3,000 iteration. So from 2,000 to 3,000 iteration, my value dropped from 5.3 to 5.0. So not a huge lot of improvements on my viSNE map. However, with one additional 1,000 iteration, it dramatically increased my runtime. So this is an indication, when is a good time to stop the iteration, what is a good iteration to choose from for this particular data set. We can easily just use a 1,500 iteration for this data set where I have a lot of markers and a lot of events. That will reduce the runtime a lot. So here's the information about the KL divergence value. So now let's go to the gating of your viSNE map. So this shows you the viSNE map on the viSNE1 and viSNE2 in the gating page. What we can do here is to use those gating tools to generate gates on the map. So in this case, we also have a coloring channel. Other than X and Y, we have a coloring channel that uses a third channel to illustrate the marker expression on the map. If I choose a CD19, you can view the CD19 positive expression on the viSNE map with each sample. So you can navigate to different sample and visualize the marker expression that way. Or we can use another marker like CD14 marker here and visualize the CD14 marker on different samples. You can use those gating tools to gate on the CD14 positive events. So I will use a V to indicate the viSNE, it's the viSNE monocytes or viSNE CD14 positive cells. viSNE CD14 positive, says okay, and that will shows you the CD14 positive gates on the viSNE map. We can now click apply and return to bring to the illustration page to visualize the viSNE map from there. So with this illustration, I show the channel as the viSNE channel, and then 2 time points, one CD45 positive population, and one patient here. So you can, in order to simplify the visualization, we can just show 1 time points and 1 patient here. So I'm going to update the illustration that way. So what we are viewing is this patient at this time point of the viSNE map illustrated by each different channel. So we are viewing the map by each viSNE channel. So each channel will have a V, and behind we can view the CD14 positive channel expression and all the other CD57 population here, CD19 channel expression here. So by viewing the map with one channel at a time, we are able to identify all the viSNE island. And remember, we just made a CD14 positive gates. So if we go down here, we can show the gates on the map. So now you can see the percentage of the CD14 positive for this particular patient at this time point. So we can definitely add more time points here to do a comparison. So now we are visualizing the different 2 time points on this same patients, and we can see this each marker expression compare side to side. We can also add a little more patients here. Add one more patient. We, instead of viewing comparing the time points, we can also comparing the by patients. So we can move this patient individual ID to the rows and then update. So now we have the viSNE map side to side as 2 patients side to side, and viewing. You can want this is the table is at time points. So the first one is acute, and the second table is the convalescence time points. So there are different ways you can build up the report. Other than all showing the channel that were used at the viSNE, we can also showing channel that wasn't included in the viSNE map, but we can definitely show them on the map afterwards. So we can add here just to show 2 additional channels PDL1. I can move them to the top. Move them to the top here, and then we can update the illustration to show those channels on the map as well. So now we are viewing the CD1 expression and the PDL1 expression for those 2 patients. We can also switch the dimensions to view the comparison between 2 time points instead of each individual patient. So now we are viewing them as CD1 expression and PD1 expression between 2 time points for each individual patient. Those are the expression. Another technique we showed earlier in the slides is to overlay the populations on the viSNE map. So we can do that as well. I'm going to select less channels to be less confusing. So 2 time points, maybe just 1 patient in this case. And the population, we can choose the manual gates we have. So we can choose a CD 16 positive NK cells. We can do CD3 cells. We can do CD19 cells, CD4 and 3, and then we can do CD19s. So in order to overlay, the population need to move to the second position, and we will choose the color by instead of channel. We are choosing the overlay figure dimension, so this population will be an overlaid dimension. And then we now can update the illustration here. So now what we are viewing is the viSNE map of one patient, showing those are the manual gates we have. So we are overlaying the manual gates on top of the viSNE map. And this is the same patient at the recovery phase, where also the manual gated is overlaid on the viSNE map. So this is the overlay. There are a few other options where you can do heat map as well. So we can choose the plot type as heat map from the down below. We need to, for the heat map, we need to choose a few channels. So I'm going to select all the CD markers here, or all the, maybe all the viSNE markers by typing the V's, select them all, and done. And then we can population, just choose, yeah, this looks good. Time points, one patient. And down here, what we need to change is the, we can also view the panel and the channel, and choose some of the median and raw transform. First the columns, minimum, and then view the heat map this way. So this is a heated map that's showing the populations on the row and the channels on the column, to have a very visualized heat map of your data. Again, the export will be very easy for you can copy and paste it to other PowerPoint. So this is a brief introduction to the viSNE algorithm, and I'll be happy to take any questions now.
All right, that was awesome. Thank you, Chunjin, very much. And thank you all for attending. So I'm going to introduce our panelists first and foremost. So you guys, I'm going to direct the questions to them. We do have a lot of questions, excellent questions, if I may add. So you all you've heard Chunjin, so she's the application scientist. On the call, we also have our data scientist, Steph Troff. And she'll be able to answer a lot of the questions. We also have Sarah Schuett, who is the East Coast Technical Sales Specialist, and she'll be able to answer a lot of really good questions as well. So what I'm going to do here is I'll read out the questions from the beginning. And as I said previously, if we don't get to them, I promise that we will email you the answers. And there was a lot of really good questions, so we'll go through them. So the first question that we have here is there an algorithm to determine an optimal iteration number for viSNE analysis, or is this simply done through trial and error? So I'm going to let Chunjin answer this question.
I'll actually jump in, Alina. Hi, everybody. This is Steph. Thanks so much for joining us. Yeah, so that's a great question. And with the original viSNE and viSNE, there is no intrinsic calculation within the algorithm itself for the best number of iterations. But because you're running in Cytobank these algorithms in the cloud, you can set up as many runs as you want. So I like to actually iterate the number of iterations. We start with the default, and we give some guidelines, some recommendations for the number of iterations within the support documents. If you go to support.cytobank.org, some recommendations based on things like the number of events and the number of channels. I personally always like to start out just using the default of 1,000 iterations, so I can get a good, quick first look at my data. And then from there, if I like what I see, I'll set up a run with more iterations, so I don't have to wait very long to get a first look at my data.
Thank you, Steph. All right, we're going to continue on. So the next question is, how is downsampling performed? Does it take a portion of each merged file, or does it take in the order of cells in the merge?
So Steph? I'll answer that as well, thanks. So when you specify that a certain number of events that you want to select within your viSNE settings, and that is less than the total events, random downsampling will be done. So it's not based on the order of events in your files. It'll be random.
Okay, perfect. Next question is, how do you assign the identity of the various clusters once a viSNE map is generated? Is this automated? Steph, do you want to take that as well?
Thanks. Yep, so Chen Jun, prior to running viSNE on these data, had made a number of manual gates based on what she knows. So it was manually done. So viSNE will not automatically assign those populations for you. It will show you the marker expression as she showed with the color coding by each channel, but it won't assign identities for you. That's up to you. If you come to check out our session on Thursday, we'll show you how to run FlowSum after this step, after you've done viSNE, and that will divide your samples into clusters. So it will define the populations, but it still won't give you a name for those. You still have to go through and incorporate some of your prior knowledge and you and your colleagues' knowledge or publications knowledge to decide what those cells should be called.
Perfect, thank you very much. Okay, I'm just going to, a really good question. What is the difference between viSNE and UMAP? And are we planning to integrate UMAP into the Cytobank? So I will let Steph answer this one as well, what the difference is, and I can comment on the integration of UMAP into Cytobank later.
Okay, great. That's a great question. So UMAP, if you're not familiar, is another dimensionality reduction tool. Oftentimes, people will run both a version of TISNE like viSNE and UMAP and see how they compare, because they'll give you they can give you slightly different messages about your data. But so in general, say consensus is that a lot of the time UMAP runs faster than viSNE, and it can be better at representing the global distances. So on a UMAP map, points that are really far from away from each other will mean more than what it means on a viSNE map. Whereas on the flip side, I think for a lot of data sets, viSNE is better at preserving that very local structure, that structure of events that appear close to each other. And you can, if you want to run UMAP, Alina will talk more about if and how we would integrate that into Cytobank, but you could run that using the Cytobank API if you can use R, or the command line, and pull it back into Cytobank and compare them side by side.
Okay, and with that said, we do have a roadmap for integration of new algorithms into the Cytobank, and so can't say exactly when, but there are things on the roadmap that will happen. So there will be new algorithms coming. Thank you. All right, another really good question. How many sample files can you run at one time for viSNE?
Salina, I can take that question.
Mm-hmm.
So I didn't mention during the app demo that in Cytobank, we can take, for the premium side, we can take up to 1.3 total events from all files. That's quite a lot of events that you can run. If you do run large data set, we also have some tricks that you can do to include as many files or as many events as possible. So you can gate down further, or you can use split files by population to include more samples and more events. And in an enterprise account, you can run up to 2 million events for the viSNE.
Perfect. Okay, we had a couple of questions that came about with respect to why we didn't include CD45 in our initial gate, or why we didn't include forward scatter, side scatter in the channel when we ran the viSNE. Chinjun, do you want to tackle this one?
Yeah, definitely. So for the viSNE of the channel selection, you do have the flexibility to decide which channel to be included in the viSNE map. So the general guidance is to choose the marker that can help with the subset separation. So mostly that's canonical markers, the lineage marker, then help to separate the viSNE island. And for some of the activation marker, they can express on different subsets that would not help to separate the islands. So that's why we would recommend not include them. The less marker you have, the faster viSNE run gonna do. And the other consideration I mentioned earlier is where we do not want to mix the scale type. So for viSNE, we recommend use ArcSine H scale to run the viSNE. Otherwise, the log not going to pass through, so viSNE most likely will fail for the log scale, as it will constantly looking for negative values for the algorithm. So that's why we said do not include the linears with the ArcSine H marker in the same run. And then for the CD45 marker, because that was a marker in that data set, we use CD45 marker to do the pre-gating. So all the cells that included in the CD45 in the viSNE run, that's already CD45 positive. So that's why even you include the CD45, all the events will be positive. And for the viSNE run, it will still try to identify low and high relatively, right? So you'll get a CD45 map with some of the lower expression, some of the higher expression. So it does not really make much sense. That's why if you have used a marker that's already being used for pre-gating, do not include in the viSNE run. I hope that's explains some of the marker selection guidance we talked about earlier.
That's perfect. Okay, another very good question. So if we are using a seed function, does this mean that we can generate an analysis template for longitudinal experiments for future use? So can we copy a seed function so that we can apply it to additional, so if we're doing a longitudinal study, such as in the clinical trials, can we utilize the same seed function and basically replicate the same viSNE analysis?
So yeah, I can take that question. For the seed, there's some setting we have to be careful, is that the seed will work for the initial seeding, right? So the seed only works when you're working with the same data. That means the same data type, the same panel. So if you're running a viSNE on a different time points, so day 1 and day 2, copying the seed may not work as well. So in order to compare longitudinal study to run viSNE, we still recommend you to run viSNE on all files in one run. So that will go back to the original question, where how many events can you take, right? If you're run large studies, so we will recommend you to include all files by doing some pre-processing to make sure you can include all those files with different time points and do the viSNE comparison. But viSNE is more exploratory, so we can still use that. You don't have to include all the events. As Stephanie will talk about, for the clustering algorithm, we can use other algorithms for more quantitative results. So the cell number shouldn't be a limiting factor for the viSNE run in that case.
That's a perfect answer. Thank you so much. All right, so we're going to get a little more technical again, and some of our users would like you to explain what is the theta parameter and what is the difference between theta and perplexity.
Excellent. I can take the question, and Steph, if you have more comment, please add. So for the theta, it's a value that measures the difference between, so viSNE, that's the algorithm, that's the original viSNE algorithm, and that's Barnhart implementation. So most of the viSNE algorithm out there using the Barnhart implementation, and the theta, if you set the theta to 0, that means the algorithm viSNE use exactly the same viSNE algorithm, which is the original one, quite slow. So it does the n pair calculation, so it's quite slow. And you set the theta higher, so it will be the modified Barnhart implementation will be much faster. However, we do not want to set the theta too high. So the default value here is 0.5. In most of the case, it works perfect for majority of the data set. However, there are some situations where you run large data set and viSNE just cannot go through. In that case, you can increase the CETA a little bit higher, but not all the way to the 1. So the CETA, it can be modified, but not too much. It's the measurement of the bound heart implementation of your algorithm in that case. So yeah, in majority of data set, just leave it as default.
Perfect. All right, another very good question. So which parameters would you play with, or would you basically change first to resolve, to get in, if you aren't resolving the populations? Would you go through iteration or perplexity?
Yeah, that's a great question. So it depends on your data set. In most of the case, the perplexity 30 works really well. So I will work with the iteration first. So work on the iteration to, if you have, in general, you can run 1,000 iteration per 100,000 events, and then increase based on there, from there. And so iteration, in most of the case, iteration can help resolve the map really well. And you can consider to increase the perplexity if you have a very complicated panel and have like lots of events where it's a lot of events from the total cells. So we can definitely adjust the perplexity. However, iteration, I will always recommend to work with the iteration first.
Perfect. Okay, more technical questions. What is KL divergence, and which range should it be?
Great question. And to be honest, I don't know what is KL divergence. So KL divergence actually it's a value. So put it in a simple way, it's a value that you can use to measure the matrices of the high dimensional data versus the matrices of the low dimensional data. So the more similar of those two, the lower KL divergence will be. And in most of the case, if it goes to the single digit, we would think it's a good viSNE map. So we want to be in a single digit. However, there's still improvements. In most of the case, you increase the iteration a lot more. Like for the data set I use, increase additional 1,000 iteration. My KL divergence value just dropped like 0.3 points. So that's not a lot of improvement. So it's already bottomed out. You can see if you plot the KL divergence on the plot, you will see a drop significantly at one point, and then it kind of plateau, bottom out. So we would think, once they drop to the single digit, depend on how the requirement of the viSNE resolution. Once it's single digit, if you're looking for some of the rare population and tiny subsets, then you can run additional iteration. Otherwise, for major subset separation, once they drop to the single digit, you should be good enough.
Perfect. Thank you very much. Okay, so another very good question, and we did cover this in a way, but I'm just going to get you to reiterate. How do we decide what seed to assign if we're looking for consistent results?
So the seed actually it's a random value. So we do not assign the seed, or we do not recommend to assign the seed for a particular run. So in general, we just let the viSNE algorithm generate a random value. And in order to run the same data set with a different iteration setting, you can definitely copy the seed from the first run and then start the new viSNE run with the same setting, but increase the iteration from 1,000 to 3,000 and use the same seed, so that for the second viSNE run, you can generate a very similar viSNE island map comparing to the previous run. That's how we can use the seed to control the viSNE island.
Perfect. Okay. All right. Sorry, guys. You have so many wonderful questions, and I'm trying to get through all of them. We have 4 minutes left. Okay, let's see. So this is a follow-up question. Chinjun has already touched upon this. And so the question, I'm just gonna reiterate it here. Which algorithm will allow us to do the longitudinal experiment analysis in Cytobank? And Chinjun has already basically said that viSNE is an exploratory analysis algorithm. And so if you want to do longitudinal, Steph will go into more detail on FlowSum, and we also have a webinar session on, oh my goodness, I'm blanking out.
On Citrus next Tuesday.
Citrus, thank you so much. Yes, that's right. So please join us in those sessions, and they will be definitely very useful algorithms for longitudinal experimental analysis. Okay. The next question, how trustworthy is low expression values of CyTOF data in terms of CD19 dim cells demonstrated in the viSNE? Is that true staining or more likely background?
I'll take a crack at this one.
Okay.
So mass cytometry data versus fluorescent cytometry, if you use it, you know that it has a lot less background. So with fluorescence, your negative values, your negative cells that don't express a marker will usually still have some value greater than 0, right? But with CyTOF data, they don't. Usually negative is 0 in CyTOF data. So generally, mass cytometry data, any values greater than 0, I think are meaningful.
Excellent. Okay, thank you, Steph. All right, let's see. Can we regroup patient samples into other categories in Cytobank, or do you group the patient samples on Cytobank?
Yeah, I can take this one. So this is about the sample tagging in Cytobank. We didn't have time to show the sample tag in the last session. So basically, you can tag or annotate your sample in Cytobank whatever way you want. You can tag them at per patient or per time point or per condition, per treatment. So in order to do this, you can just go to the sample tag and assign tags to each sample. You can easily find those instructions on our support site.
Perfect, thank you so much. And I'm gonna get Sarah to answer this question, which is, can the plots be exported as vector files?
Yeah, so we have a few different ways that you can export plots out of Cytobank. You can export them as the SVG data files, and I believe that's just by, and Steph and Chinjun, right-clicking on the plot to save it as an SVG file, but it is available out of Cytobank.
Perfect, thank you very much. Okay, guys, well, we just have 1 minute left. And in this 1 minute, I just want to thank you all, to all the participants, as well as our panelists. We really are enjoying giving you these webinar educational series. So please join us next Thursday for the forthcoming webinar. So thank you all and have a wonderful day. Stay safe and healthy. Bye-bye.
Thank you. Bye-bye.